Good afternoon. My name is Abhinav Latif, and I'm joined here with Daniel Cruz, Chris Garfalo, and Carlos Zabalara. Today, we are going to be presenting on protein structure and synthesis. We're going to start off with an intro here and show you an example of a quaternary protein structure, which is shown to you right here. Proteins are essential macromolecules with complex structure and that enable them to carry out various functions within the cell, including replicating DNA, catalyzing metabolic reactions, transporting molecules, and forming muscles. The structure of a protein is closely linked to its function. In this presentation, we're going to go over the four protein structures, transcription, translation, and effects of genetic mutation on protein function and structure. And then we're going to go over two specific proteins, insulin and hemoglobin. First, we're going to stress the four levels of protein structure. The first being primary. As you can see on the top picture to your left, we have a primary structure there, which is a long strand of what seems to be beads, which is held together by peptide bonds, a chain of amino acids. The primary structure of a protein is a sequence of amino acids that make up the protein chain. The sequence is determined by the genetic code and is crucial to the protein's function. Even minor changes to the sequence can significantly impact the protein structure and function. At the bottom left picture, you can see here the two structures, the helix and the beta pleated. The helix in which it coils and the pleated in which it folds in on itself. The secondary structure refers to the local folding and twisting of the protein chain into specific shapes, such as alpha helixes or beta sheets.
There is two other levels of protein structure. They're named tertiary structure and quaternary structure. The tertiary structure is the overall three-dimensional shape of the protein, which is determined by numerous factors, including the interaction between different amino acid side chains, and the presence of disulfide bonds. On the other hand, the quaternary structure refers to the arrangement of multiple protein chains to form a functional complex. Protein synthesis. In the protein synthesis, there is two primary stages, named transcription and translation. The transcription is the mechanism by which DNA is transformed into messenger RNA, which carries the genetic information to the ribosomes. The ribosomes utilize the mRNA to initiate protein synthesis through the process of translation. During translation, the ribosomes read the mRNA and leverage to construct a sequence of amino acids that form a protein chain. The genetic code is translated into a specific sequence of amino acids that constitute the protein. As stated in molecular biology, proteins that bind to transfer RNA are required as assembly factors for protein synthesis on the ribosome. This is discussed in chapter 6. In each case, an important biological activity is controlled by a change in the proteins.
So now I'll be talking about protein synthesis and genetic mutations. So we know that every genetic code has, well, each amino acid sequence is genetic code. They're made similarly like this into helixes, as we can see in the bottom picture. However, if we take pieces out, then we can cause that mutation. The structure and function of these proteins can be substantially affected by these mutations. Any alterations in these amino acid sequences can lead to mutations and modifications. Furthermore, mutations may influence the regulation of protein synthesis, resulting in irregular protein expression levels. For instance, some mutations that affect the synthesis of hemoglobin can lead to disorders such as sickle cell anemia, which is not good for the body. This disorder arises from single amino acid substitution in the beta chains of hemoglobin, which is basically what's happening in this photo down here. So now I'm going to be talking about one of our proteins, which is hemoglobin. It is a complex protein accountable for the transportation of oxygen throughout the body. The composition of hemoglobin consists of 4 protein chains, 2 alpha chains and 2 beta chains. The primary structure of each chain is very distinct, and each has a particular function in the overall role of protein. Hemoglobin's quaternary structure plays a critical part in its function, as it enables the protein to bind to oxygen and carry it to different parts of the body. By binding reversibly to oxygen gas through its iron atom, heme enables hemoglobin to pick up oxygen in the lungs and release it into the tissues. The flexibility of hemoglobin structure allows it to change its conformation and bind or release oxygen based on the needs of the specific body parts.
So next thing we'll be talking about is insulin and different types of proteins in the body. Insulin is a protein hormone regulating glucose levels in the body. It is comprised of 2 protein chains linked by disulfide bonds. The primary structures of each chain is unique, and their collaboration results in the formation of functional insulin. The structure of insulin plays an important role in its function by facilitating its binding to specific receptors that regulate glucose and uptakes by cells. Insulin synthesis occurs in the pancreas and is released in response to increased blood glucose levels. Upon insulin binding to its receptors on a cell surface, it initiates a signaling cascade that facilitates the uptake of glucose from the bloodstream into the cells. This process is crucial for maintaining glucose homeostasis and defects in insulin synthesis or signaling. This may result in diseases. So to summarize, or to be in conclusion, the proteins are intricate macromolecules that carry various functions within the cells. Their structures are closely tied to their functions, and producing protein is a complex and highly regulated process. The floor levels of protein structures include primary, secondary, tertiary, and quaternary. Determines their shape and overall function. These stages of transcription and translation are crucial for protein synthesis, and genetic mutations can have a significant impact on protein structures as well as function. Hemoglobin and insulin exemplify 2 proteins with these unique structures and roles, and defects in their synthesis or signaling may result in different types of diseases. Comprehending protein structures and synthesis is fundamental to understand the organization of living organisms and develop new treatments for protein-associated ailments. So this is our works cited. This is all the information that we've gathered